a slot with us anytime in case you want an extra guidance and support from us. Then we have three different subscription plans at Oracle: basic, standard, and premium. The basic is for 3 months and it comes at 15,000. The standard is for 6 months and it comes at 30,000. And the premium is for 12 months, which will cost you 50,000. Then you can have additional 20% off on any of these subscriptions, as I told you, by using my promo code as displayed here on the screen. That is Dr. Beenish 20. Then apart from that, we have internal exam marathon series right now going on. So introducing you a little bit to that series. This series is specifically curated for your university exam. So you have rapid revisions of all the important topics, whatever questions are being frequently asked in the exam. So we're trying to cover all of them quickly in a rapid revision mode. Then all volatile facts will be covered for quick revisions, and you have MCQs so that we can assure you top-notch grades in your exams. Okay, so today we'll be talking about tryptophan metabolism. So beginning with that, a little introduction about tryptophan. Talking about the structure of tryptophan, it has an indole ring. So now what is an indole ring? Your indole ring, it consists of benzene ring, fused with a pyrrole ring. So they together form an indole ring. Now, what is a pyrrole ring? It is a five-membered ring with nitrogen. So this is a benzene ring, and when this benzene ring, it is fused with a five-membered pyrrole ring like this forms what is known as your indole ring. So this indole ring is a characteristic feature of tryptophan. This is present in tryptophan. So it is the only amino acid with two rings in its structure. Okay. So you might get in an MCQ like, which of the following amino acids they have two rings in their structure? So your answer will be tryptophan. Then it is an essential amino acid. It is aromatic and it is both ketogenic and glucogenic. So if we talk about the metabolism of tryptophan, its catabolic fate, it includes both ketogenic fate and glucogenic fate, and anabolic fate, that is, it is involved in the synthesis of niacin, serotonin, and melatonin. Just a second. Okay. So if we talk about the catabolic fate of tryptophan, it is also known as kynurenine anthranilide pathway. I think only half of my screen is visible. Okay, yeah. Sorry for the disturbance. Okay, so we were talking about the metabolism of tryptophan. So its catabolic fate, it includes both glucogenic and ketogenic pathways, and anabolic fate, it is involved in the synthesis of niacin, serotonin, and melatonin. Now moving on to the catabolic fate of tryptophan, it is also known as kynurenine anthranilite pathway. So what is happening in this? That the 11 carbons, as in when we study the pathway, you will get to know. But in a summary, let me summarize it to you that the 11 carbons of tryptophan, they are oxidized into, you can see, formyl group. Then you can see here alanine, and then carbon dioxide molecules, they're removed as carbon dioxide molecules, and then acetoacetyl-CoA is formed. So the 11 carbons of tryptophan, they are oxidized as 11 carbons of tryptophan are oxidized as formyl group, which is 1 carbon. Okay. Then you have alanine, which is 3 carbon. Then you have acetoacetate, which is 4 carbon, and then the remaining 3 carbons, they are removed as 3 carbon dioxide molecules. So the remaining 3 carbons, they are removed as carbon dioxide, as 3 carbon dioxide molecules. Okay. So that was briefly about the tryptophan catabolism. So this formyl group, which is, so this makes up, if you add all these carbons, it will come to 11. You can see it for yourself. You add this 1 to this, you add this 3, so it becomes 4, and then you add another 4, it becomes 8, and then you add 3 more carbons, so it becomes 11. So you see the 11 carbons, that is how they are oxidized. So this formyl group that is formed, it enters the 1 carbon pool. So alanine, it enters the glucose pathway. So acetoacetate, it enters the ketogenic pathway. Okay. So now let's take a look at the stepwise reactions. Okay. So your tryptophan, it is changing into N-formyl kynurenine. The enzyme is tryptophan pyrrolase. Here, tryptophan pyrrolase. This is a dioxygenase and hemoprotein. That is, it is a heme containing enzyme. Okay, so what is formed? N-formyl kynurenine is formed. Now, from this N-formyl kynurenine, this formyl group, it is given to tetrahydrofolic acid to form what? Formyl tetrahydrofolic acid. As you can see, this tetrahydrofolic acid, it enters 1 carbon pool. Now, this N-formyl kynurenine, after giving its formyl to tetrahydrofolic acid, it becomes kynurenine. Okay, now this kynurenine, what is happening? It is getting, you can see a hydroxyl is being added to it. So it is getting hydroxylated to form 3-hydroxykynurenine. Now this 3-hydroxykynurenine, it is then changing into 3-hydroxyanthranilic acid, and in this reaction, you can see what is liberated. Alanine is given out, and PLP is required for this reaction. And what is the enzyme? The enzyme is kynureninase. Okay, this kynureninase is the enzyme which requires PLP. Okay, so this requires PLP. So in case of vitamin B6 deficiency, what will happen? This reaction will not take place because your kynureninase, it is dependent on PLP, okay? So this reaction will not take place, so your kynurenin will increase and 3-hydroxyanthranilic acid, it will decrease, okay? Now this 3-hydroxyanthranilic acid is what goes into NAD pathway. What is NAD pathway? That is, it is the NAD is the active form of niacin. Okay, so what will happen? So NAD will be decreased in case of vitamin B6 deficiency. Okay, and what happens? This 3-hydroxyanthranilic acid, then by further reactions, you can see decarboxylations taking place. Ultimately, what is formed? Acetoacetyl is formed, which enters into ketogenic pathway. So that was about the catabolic fate of tryptophan. So you've seen this alanine, which was given here in this step. This is entering the glucogenic pathway. Ultimately, the end product that is formed, acetoacetyl-CoA, it is entering the ketogenic pathway. And also the intermediate 3-hydroxyanthranilic acid that is formed, it is entering the NAD pathway. Okay, and in the NAD pathway, it is a multi-enzyme pathway. Multiple enzymes are needed. So the most important enzyme, that is the rate limiting enzyme in this NAD pathway. The rate limiting enzyme is QPRT, that is quinolinate phosphoribosyl transferase. Okay, so this is the rate limiting enzyme. Enzyme of NAD pathway. So I'll just quickly repeat this for you. So tryptophan, it changes into N-formyl. In your tryptophan metabolism, you are getting a formyl group, which is entering the 1-carbon metabolism. Then you're getting alanine, which enters the glucogenic pathway. You're getting acetoacetyl-CoA, which is entering the ketogenic pathway. And then you have the intermediate 3-hydroxyanthranilic acid, which is entering the NAD pathway. Okay, so these are the important points that you need to remember. So I'll summarize the important points from the point of view of MCQs that you should be remembering. That is, your kynurenine is an intermediate of tryptophan catabolism. Okay. So after you've studied the metabolism of all the amino acids, it will get really confusing. So you should like bullet these points down, like even a simple MCQ that kynurenine is involved or is an intermediate in the pathway of which of the following amino acids, and you have multiple options. Okay, so sometimes even as direct questions, such direct questions will also confuse if you're not very thorough with the facts. Okay, tryptophan pyrrolase, also known as tryptophan oxygenase, as I told you, it is a heme containing enzyme. Amino acid which donate formyl group to 1-carbon metabolism is your tryptophan. Again, an important one-liner. And vitamin K required for kynureninase is PLP. So in case of vitamin B6 deficiency, is excretion of xanthurenic acid in urine. So if you take a look here at this step, what is happening here? In case of vitamin B6 deficiency, so your kynureninase will not be working. So what will happen? Your kynurenin will increase. The level of kynurenin will increase. So it will form what? Xanthuric acid. Okay. So there will be secretion of xanthuric acid in the urine. Then synthesis of NAD plus is also decreased in case of B6 deficiency, leading to pellagra-like symptoms. Okay. Now talking about the anabolic fate of tryptophan. So as we said, it is involved in the synthesis of niacin, serotonin, and melatonin. So we'll take each of them one by one. So your serotonin synthesis is take place in the argentifin cells, which are present where? In the intestine, mast cells, platelets, and brain. So let's have a look at the reaction. So what is happening? Your tryptophan, it is changing into 5-hydroxytryptophan. So you can see what is happening. Hydroxylation is taking place. You can see a hydroxyl has been added in this position. So what is happening in the first step? Hydroxylation has taken place. And if you look at the mechanism of this reaction, you'll find it to be very similar to phenylalanine hydroxylase. Isn't it? Because your hydroxylation is taking place, then also there is requirement of tetrahydrobiopterin, which is being reduced to dihydrobiopterin, and then your dihydrobiopterin is again being regenerated with the help of NADPH. So this is very similar to phenylalanine hydroxylase. Okay, so your tryptophan by hydroxylation, it changes to 5-hydroxy tryptophan, and the enzyme is tryptophan hydroxylase. Now what happens? Decarboxylation is taking place. You can see a carbon dioxide molecule is being removed. So decarboxylation takes place. Now what happens? Via decarboxylation, your 5-hydroxytryptamine, which is known as serotonin, is formed. Now this serotonin then gets acetylated. So what is taking place? As you can see here, acetylation is taking place. So your serotonin gets acetylated to form acetyl serotonin, and then what happens after acetylation? Methylation is taking place. So for methylation, what is being used? SAM is being used, which is S-adenosyl methionine, and it is derived from methionine. Okay, so your S-adenosyl methionine, which is the methyl donor, it is changing into S-adenosyl homocysteine. So after your acetyl serotonin, it is methylated. What is formed? Melatonin is formed. So this forms an important MCQ, that which amino acid is required for conversion of your serotonin to melatonin. So what is the amino acid required for conversion of serotonin to melatonin? Your answer will be methionine. Okay, because for methylation, what is needed? S-adenosyl methionine is needed, and SAM is derived from methionine. And also, your serotonin, it is degraded by an enzyme known as MAO, which is monoamino oxidase. Okay, so this MAO, it degrades serotonin to 5-hydroxy indole acetic acid. So what happens in case of depression and all? If you want to increase the level of serotonin, what will you give? You will give MAO inhibitors are given. Okay. So MAO inhibitors are given in case you want to elevate the mood. So for mood elevation, MAO inhibitors are given. So that was about the formation of serotonin and melatonin from your tryptophan. So your tryptophan first gets hydroxylated, then it gets decarboxylated to form serotonin. Then that serotonin is acetylated and then methylated to form melatonin. Now moving on to the functions of serotonin. It is an important neurotransmitter in the brain. It is involved in temperature regulation. It is involved in mood elevation and increases gastrointestinal motility. So mood elevation, how? It is used as an antidepressant. Because in patients of depression, what is low? Your serotonin levels are low. In patients of depression, serotonin is decreased. Okay, and what is the function of melatonin? So your melatonin is synthesized in the pineal gland, and it is an important neurotransmitter and important in biological rhythms, that is the sleep and wake cycle. So your sleep pattern, it is decided by melatonin. Now moving on to the clinical correlations. So first is carcinoid tumor. It is a tumor of argentifin cells and also known as argentifinoma. And what are argentophin cells? We've just discussed that they are cells of the GI which produce serotonin and are necessary for GIT motility. So your argentophin cells, they are cells of the GI which produce serotonin and they are necessary for GIT motility. Okay, so your carcinoid tumor, there is tumor in the argentophin cell. So what will happen as the argentophin cells are producing serotonin? So what will increase? There will be increased synthesis of serotonin. So obviously when there is increased synthesis of serotonin and we say that serotonin is important for GIT motility. So what increased serotonin will cause what? It will cause diarrhea. There's increased sweating. There is fluctuating hypertension, and then also what is seen, there is cutaneous flushing. And what will happen when there is increased synthesis of serotonin, it will lead to decreased niacin synthesis. So there will be flagra-like symptoms. Now, cutaneous flushing, talking about that, there are two reasons for that. Firstly, due to excess serotonin, which acts as a vasodilator, so there will be cutaneous flushing because of vasodilation due to serotonin. And the other is that in this particular tumor, there's increased tachykinase, which also causes cutaneous flushing. So one is because of tachykinase and the other reason because of vasodilation. So that was briefly about carcinoid tumors. Now talking about the diagnosis of carcinoid tumor, there is increased serum serotonin. As we all know, there is a tumor. So what will happen? It will produce more serotonin. So your serotonin level will be increased, and then the other is increased 5-hydroxyindole acetic acid in 24-hour urine. Obviously, if there's increased serotonin, there will be increased degradation. So 5-hydroxyindole acetic acid will also be increased. Talking about the other clinical correlation, which is Hartnupp's disease. So it is named after the first family in which it was identified. There is defect in absorption of tryptophan and other aromatic amino acids from intestine and renal tubules. So amino acids are excreted in the urine. What is seen? A blue diaper is seen. So it is saying that the absorption is defective because there is defective transporters, okay? And where is it taking place? In the intestine and in the renal tubules. Let me explain you this with the help of a diagram. Consider this is your intestine. So it says there is blue diaper. So what is the reason behind this? Let's see. So your tryptophan, it cannot be absorbed. Okay, this absorption is not taking place because the transporters here, they are defective. Now what happens? This tryptophan, it is acted upon by bacterial enzymes in the intestine. And what is formed? Indole compounds are formed. Like your indigo blue and indican. Now these indole compounds, they enter systemic circulation. Then from there they go to renal circulation. And then it is excreted in urine. So your indican or indigo blue, it is excreted in urine. Okay, and this is what contributes to blue diaper. Okay, because they're blue in color. So in Hartnupp's disease, you can see there is blue diaper. Then as tryptophan is not entering systemic circulation, there is decreased tryptophan in cell, which leads to decreased serotonin and decreased niacin. So obviously as your There's defective absorption of tryptophan, so there will be deficiency of tryptophan, and tryptophan is involved in the synthesis of serotonin and niacin, so they will not be formed. Most common symptom of Hartnupp's disease is cutaneous photosensitivity because of decreased niacin. Okay, so decreased niacin can cause pellagra. The 4 Ds of pellagra we all know, it's diarrhea, dementia, dermatitis, and death. So there is photosensitive dermatitis. Photosensitive dermatitis. So, so the area that is exposed to the sun that develops rashes and lesions, especially the area around the neck. Therefore, it is known as Castle's necklace. So the exposed area to sun, it develops rashes and lesions. Okay. Then you have neurological manifestations. Obviously, why neurological manifestations? Because your serotonin is a neurotransmitter. So what will happen? There will be intermittent ataxia, white-based gazed. Okay, so there is loss of control between the arms and the legs. There's no coordination, and most of the time, it is symptomatic of Hartnupp's disease. So that was briefly about Hartnupp's disease. The important thing that your tryptophan absorption is defective because the transporters are defective. From where? From the intestine and the renal tubules. Then there's blue diaper. Why? Because the tryptophan in the intestine, which is acted upon by bacterial enzymes, which form indole compounds, and they are finally entering into systemic and renal circulation and excreted in urine. Then what is happening? Because of decreased tryptophan, there will be decreased synthesis of niacin, which will lead to pellagra-like symptoms. And then you have the manifestations of pellagra, that is the 4Ds, diarrhea, dementia, dermatitis, and death. Now talking about the diagnosis of Hartnupp's disease. So the test is known as Ombermere test, which is for indole compounds, and treatment includes supplements with niacin as your niacin synthesis is decreased, and minimum exposure to sunlight as there is photosensitive dermatitis, cutaneous photosensitivity. Okay, then talking about the biochemical defect. Now the defect is in the transporter which transports tryptophan and other neutral amino acids in the circulation. The transporter that is defective is both one, and it is coded by the gene SLC6A193. Although the name of the gene is not very important, but should know that the transporter is both one, which is defective. Okay. So talking about Ramon syndrome, it is also known an amino acid transporter defect, but the difference is here the both one transporter is defective only in the intestine. Unlike Hartnupp's disease, where it was defective both in the intestine and the renal tubules, here the both one is defective only in the intestine, and it is known as blue diaper syndrome. So now don't get confused. The blue diaper is also seen in Hartnupp's disease, and it is also seen in Drummond's disease. But the name that the blue diaper syndrome name, it is only given to Drummond syndrome. So if the question says the blue diaper syndrome is also known as, and in the options you have Hartnupp and Drummond both, so your answer will be Drummond syndrome. Okay, because as the name is only given to Drummond syndrome. So that was about tryptophan. Now let's talk about glycine and serine. Okay, so talking about glycine and serine metabolism, let me just introduce you to glycine first, the structure of glycine and its properties, general properties. So your glycine, it is a non-essential amino acid, that is, it is not taken through diet, it can be synthesized from various substrates in the body itself. Now it is not asymmetric. Okay, that is, it shows symmetry because we all know the structure of glycine. What is the structure of glycine? This is the carboxylic group, this is the amino group, and this is the hydrogen. So this is the constant part of any amino acid, and then here the variable R is also an H. So you see 4 different groups are not attached to this carbon. So it is not a chiral carbon, it is not asymmetric. So it is not optically active carbon, it is optically inactive. It is purely glucogenic. It is a polar amino acid and it is the simplest amino acid. Okay. So now talking about that from where all your glycine is synthesized, it is synthesized from glyoxylate, which is the keto acid for glycine. It is the keto acid for glycine. Then from the glycine synthase, then from the serine and threonine. Talking about the catabolic plate, it gives serine and the glycine cleavage system. We will talk about this in detail later in the lecture. And talking about this anabolic plate, it is involved in the synthesis of carbon 4, 5 and nitrogen 7 of purine, in the synthesis of creatinine, heme and glutathione. So let's talk about the synthesis of glycine. So it can be synthesized from serine, glyoxylate, and threonine. So talking about its synthesis from serine, so you can see if you compare the structures of serine and glycine, I'll just show you the structure of serine. So I'm drawing the constant part of serine, which is constant for any amino acid. Okay, this one is the constant part. And now this serine, as we all know, it is a hydroxyl containing amino acid. So it has a beta carbon to which a hydroxyl is attached. So this is the extra part in serine. So serine has an extra carbon and a hydroxyl, as you can see, this extra carbon and this hydroxyl as compared to your glycine. So they have to be removed. This beta carbon and this hydroxyl has to be removed. So this has to be removed and this is removed as methylene tetrahydrofolic acid, okay, because there's just one beta carbon to which this hydroxyl is attached. So the beta carbon of serine is removed as methylene tetrahydrofolic acid. Carbon of serine removed as methylene tetrahydrofolic acid. Okay, so let's see. This is your serine. It is acted upon by the enzyme serine hydroxymethyl transferase, and you can see from this serine what is also needed. Tetrahydrofolic acid is also needed, and your beta carbon is given to this tetrahydrofolic acid to form methylene tetrahydrofolic acid, and what is formed from serine now? Glycine has been formed. So this methylene tetrahydrofolic acid that is formed, this is the primary donor of one carbon group. This is the primary donor of one carbon group. And also what is needed for this reaction? PLP and folic acid. Folic acid, you can already see why is it needed. Okay, so that was about the synthesis of glycine from serine. So serine is acted upon by serine hydroxymethyl transferase. PLP and folic acid are required. Your beta carbon is removed as methylene tetrahydrofolic acid, which is the primary donor of one carbon group, and ultimately what is formed? Glycine is formed, and also this is a reversible reaction. Okay. Now, moving on to synthesis of glycine from glyoxylate, which is the keto acid of glycine. So, obviously, from the keto acid, the amino acid is formed. So, this will take place through transamination. So, what is required? Another pair of keto acid and amino acid required. So, it can either be alanine or glutamate, and it is acted upon by the enzyme alanine glyoxylate transaminase in case of alanine. And what is formed? Your amino group from alanine is transferred to glyoxylate to form glycine, and alanine itself is reduced to pyruvic acid. In case if it was glutamate, then glutamate would change into alpha ketoglutarate. And the coenzyme required is PLP. So that was the synthesis of glycine from glyoxylate. Now talking about the synthesis of glycine from threonine. So your threonine, it is acted upon by threonine aldolase, and it will split your threonine into glycine and acetaldehyde. Okay. Now let's talk about the glycine cleavage system. So it is a multi-enzyme complex. It consists of H protein and three enzymes. What are the three enzymes? They are glycine dehydrogenase or glycine decarboxylase, aminomethyltransferase, and dihydrolipoamide dehydrogenase. Okay, which is covalently attached to H protein. So the glycine via the glycine cleavage system, it is completely degraded to, as you can see, carbon dioxide, ammonia, and methylene tetrahydrofolic acid. Okay, so your glycine is completely degraded to carbon dioxide, ammonia, and methylene tetrahydrofolic acid. Okay, so what is happening? Your glycine, it is acted upon by the enzyme glycine decarboxylase. So what will happen? Decarboxylation, as you can see, a carbon dioxide is removed, and what is formed? Amino methyl group. So you're left with the amino group, the methyl. Okay, and the carbon dioxide, the carboxylic group has been removed. And what is needed for this? The coenzyme is PLP. Now what happens? This amino methyl transferase will act, and this enzyme, it will separate the methyl group and the amino group. Okay, so amino methyl transferase will act now, and it will separate the amino and methyl group. Now this methyl group, it is taken up by, as you can see, tetrahydrofolic acid to form methylene tetrahydrofolic acid. And what is left now? This ammonia is formed. Okay, and for this reaction, what is needed? Lipomide is needed, which is then changed into reduced lipomide. And reduced lipomide is regenerated back to lipomide. For this, the enzyme is lipomide dehydrogenase. So that was about glycine cleavage system. Now all these reactions, they are all reversible. Okay. So all the reactions taking place in glycine cleavage system, they are all, these reactions are reversible. Reactions are reversible. So your carbon dioxide, ammonia, and methylene tetrahydrofolic acid, they can all join together to form glycine. And then in that case, when these reactions take place in the reverse direction, which is known as glycine synthase system. It is known as glycine cleavage system when glycine is completely degraded to carbon dioxide, ammonia, and methylene tetrahydrofolic acid, which is known as glycine cleavage system. Now I'm saying that all these reactions are completely reversible. So when carbon dioxide, ammonia, and methylene tetrahydrofolic acid, they all join together to form glycine, which is known as glycine synthase system. Very simple. Okay, now talking about the anabolic patent functions of glycine. So first is keratinine. So it is involved in the synthesis of keratinine. So three amino acids are basically involved in the synthesis of keratinine. That is firstly your glycine, arginine, and methionine. So very important. Sometimes you might get asked what are the amino acids involved in the synthesis of keratinine, and the options would be all the options might be a combination of three amino acids. Okay, so you should know all three of them and not just know one or two. So apart from glycine, your arginine and methionine are also required. How? We'll just see. So what is happening? In the very first step, what is happening? Your glycine, it combines with arginine to form guanidinoacetic acid. Okay. So you can see glycine is required and arginine is required, and this is taking place in the kidneys. So this takes place in the kidney. Now what happens? Methylation is taking place in the second step. And methylation, SAM is needed. Okay. Now this SAM, we all know it is derived from, SAM is derived from methionine. So you see how the three amino acids are required, glycine and arginine, they join together to form guanidinoacetic acid. And the enzyme for this first reaction is glycine arginine amidotransferase. Now, in the second step, methylation is taking place, which is brought about by guanidinoacetate methyltransferase. And from guanidinoacetic acid, keratine is formed. And for this methylation, SAM is needed. Now, this SAM, you all know, is derived from methionine. So, you see, methionine is also needed for the synthesis of keratine. Now, this keratine, it undergoes phosphorylation by keratine kinase. Okay, so you see this ATP is required here, and what will be formed? Keratine phosphate is formed. Now, this keratine phosphate spontaneously, that is without any enzymes, non-enzymatically. Okay, this is spontaneous. What happened? It changes into keratine. Okay, so that was about formation of keratine. Now, this spontaneous reaction, which does not require any enzymes for the formation of, and one more thing. The first step, as I said, it was taking place in the kidney. The second one here, this methylation, is just taking place in the liver. And this last step, okay, this one here, the conversion of keratine phosphate to keratinine, which I said is taking place spontaneously. It is taking place in the muscles, okay? This takes place in the muscle. Okay. Now, this reaction, the last reaction taking place in the muscle, which is known as Lohmann's reaction. It is a spontaneous reaction. Spontaneous, no enzymes required, takes place in the muscle. Your keratine phosphate acts as an immediate replenisher of ATP in muscle, especially for the first 3 to 4 seconds of exercise. Okay. So, this is Lohmann's reaction. So, sometimes you might get in a question like guanidinoacetic acid is formed in. That is why I'm telling you the locations for these keratinine synthesis, because it is some partially taking place in the kidney, liver, and the muscle. Okay, so question might be that your guanidinoacetic acid is formed in, your Lohmann's reaction is taking place in. So the places are different. Okay, so you need to know which reactions are taking place where. The first one is taking place the combination of glycine and arginine, or the formation of guanidinoacetic acid is taking place in the kidney, the methylation is taking place in the liver, and the Lohmann's reaction is taking place in the muscles. Okay. Now, the other products that are synthesized from glycine, you have your heme. We'll talk about this in great detail in an entire chapter contributed towards heme synthesis. But for now, I'll briefly tell you that your succinyl-CoA combines with glycine, and then the pathway starts, and ultimately what is formed? Heme is formed. So the starting substrate, it is succinyl-CoA, which combines with glycine, and then ultimately via multiple steps, what is formed? The heme is formed. Then glutathione, we'll talk about this in great detail, how it is involved in as an antioxidant and free radical scavenging and all, and sulfur containing compounds. Okay, when we talk about sulfur containing amino acids, then C4, C5, and N7 of purine, neurotransmitter, that is in CNS and PNH, it is acting as a excitatory as well as inhibitory neurotransmitter. Then conjugating agents, your glycine also acts as a conjugating agent. We've already seen taurocholine, glycocholine were formed bile salts. They were bile acids. They were conjugated, and also for the conjugation of benzyl CoA. So you already studied about the conjugation of bile acids in cholesterol synthesis, and about benzoyl CoA we will study in xenobiotics. Okay, so your benzoyl CoA, when we talk about detoxification chapter, then we'll see that your benzyl CoA conjugates with glycine to form hippuric acid. Okay, and then glycine is also the most abundant or recurring amino acid in collagen. Moving on to the clinical correlation. So the first one is hyperoxaluria. It is of two types, primary, which is rare, and secondary, which is common. So your primary or hyperoxaluria, it is again of two types, type 1 and type 2. In type 1, the defect is in enzyme alanine glyoxylate aminotransferase. Okay, it is a peroxisomal protein target defect. And the type 2, the defect is in enzyme glyoxylate reductase, also known as glycerate dehydrogenase. So what is happening basically is there will be accumulation of glyoxylate, which later converts into oxalate and ultimately leading to oxaluria. So what is happening? Your glyoxylate, which is produced as an intermediate molecule in metabolism of hydroxyproline, glycolate, and glycine, that glyoxylate, it needs to be metabolized further. Okay, so it is generally detoxified in the peroxisomes of hepatocytes by your alanine glyoxylate aminotransferase. Now what is happening? If this alanine glyoxylate aminotransferase is defective, it will not be detoxified and your glyoxylate will accumulate. And this accumulation of glyoxylate will form what? Oxalate, leading to oxaluria. Okay. So what I'm trying to say is your glyoxylate. Where are you getting this glyoxylate from? So your glyoxylate is produced as an intermediate in metabolism of hydroxyproline, glycolate, and glycine. Now, what is usually happening, that this glyoxylate that is produced, it is generally detoxified in peroxisomes of hepatocytes by your alanine glyoxylate aminotransferase. Okay, so your glyoxylate, it is acted upon by this AGT enzyme to form glycine, but this here is defective. So what will happen? The glyoxylate will increase. See, your glyoxylic acid, in case your AGT is defective, it will increase, and in case your glyoxylate reductase is defective here, this will not take place, that it can form glycolate. Then again your glyoxylic acid will increase if this is also defective. So what will form? It will form oxalate. This increased glyoxylate, it will form oxalate, leading to oxaluria. Okay. Now that was about primary hyperoxaluria, which was of two types, type 1 and type 2. We've discussed what are the enzymes defective in both of them and what it ultimately causes. Now talking about the secondary hyperoxaluria, it mainly pertains to excess exogenous oxalate. So this In the previous type 1, we were talking about that glyoxylate that was produced in the body because of metabolism taking place of hydroxychloroquine and glycine and all. But this one is because of excess exogenous oxalate gained either through diet or due to intestinal pathologies. Okay, so some of the causes of secondary hyperoxaluria are vitamin B6 deficiency because glyoxylate to glycine is transamination reaction. So coenzyme is PLP. Now what is happening? So AGT might not be defective, but there is deficiency of PLP. Then again, if there is deficiency of PLP, your AGT will not be able to work. So because of B6 deficiency, what will happen? There will be secondary hyperoxaluria. Then from ascorbic acid degradation, you get exogenous oxalate, then ethylene glycol poisoning, and from ingestion of leafy vegetables because they are your leafy vegetables, they are also rich in oxalates. So these are the sources from where you can get excess exogenous oxalate leading to secondary hyperoxaluria. In non-ketotic hyperglycemia, it is due to defect in glycine cleavage system. Now this is because of defect in glycine cleavage system. So glycine level is increased in urine, blood and CFS, and there is severe mental retardation and seizures seen. So that was about glycine and its clinical correlation. Now let's talk about serine. So serine is again a hydroxyl containing amino acid. You can see we've discussed the structure of serine before also in the beginning of the lecture. It has just beta carbon to which a hydroxyl group is attached. It is polar and it is uncharged. It is non-essential, purely glucogenic and most common site for phosphorylation and dephosphorylation. Now, serine, it can be synthesized from glycine. It can also be synthesized from 3-phosphoglycerate. That is a glycolytic intermediate. We all know 3-phosphoglycerate. It is the major source of serine in the body, and the steps involved are dehydrogenation, transamination, and removal of phosphate. So you can see from 3-phosphoglycerate, dehydrogenation takes place. Then what is from 3-phosphohydroxypyruvate is formed. Then transamination takes place and phosphoserine is formed. And then from that phosphoserine, the phosphate is removed. That is dephosphorylation taking place by phosphatase. And then ultimately serine is formed. The reactions are not very important. Okay, talking about the metabolic functions of serine. So it is the primary source of one carbon because we've already seen how from serine when you form glycine, what was formed? Methylene tetrahydrofolic acid was formed, which was the primary donor of one carbon group. Then cysteine synthesis, phosphatidylserine, which is a phospholipid, is also formed from serine. Sphingosine is also derived from serine by combining serine with palmitoyl-CoA. Then it is also a precursor of selenocysteine. Then decarboxylation of serine, which is done by serine decarboxylase, and what is formed? Ethylamine is obtained. Okay, so your serine, when it is decarboxylated, that is carbon dioxide is removed, and for this the enzyme is serine decarboxylase and the coenzyme is PLP. What is formed? Your ethanolamine is formed, and from this ethanolamine, what is derived? Choline. Okay, then glycoproteins, that is O-linked glycoproteins, they are also derived from serine. What are O-linked glycoproteins and how are they derived from serines? So you see when proteins are glycosylated, when proteins are glycosylated, that is carbohydrate is attached to it. So you see this hydroxyl group, the H of the hydroxyl, this is removed and carbohydrate is attached to it. Okay, so this H is removed and carbohydrate hydrate group attached. So what will be formed? Your serine, which was having a hydroxyl. Okay. Now on glycosylation, the hydrogen is removed and a carbohydrate is attached. So you will have serine O carbohydrate. Okay, so this is what O-linked glycoproteins are. What are glycoproteins? To proteins, you are adding a glycosylation segment, you are adding a carbohydrate. So this is an O-linked glycoprotein. Okay, so therefore O-linked glycoproteins, they are not only derived from serines, but they're derived from all hydroxyl containing amino acids. Okay, and then this is the most common site for phosphorylation, especially covalent modifications of enzymes. So we all know that the key enzymes of pathways, they are regulated by or controlled by phosphorylation and dephosphorylation. So the active sites of these key enzymes, they have most of them, they have serine or other hydroxyl containing compounds. So these hydroxyl groups, they form the site for phosphorylation and dephosphorylation. So with this, we come to the end of our class for today. So we've covered tryptophan, glycine, and serine. If you have any queries, you can send them. So we've covered phenylalanine and tyrosine in the previous class. We're done with urea cycle. Today we've covered Hartmann's disease, Ramon's disease, tryptophan metabolism, its catabolic anabolic rate, glycine and serine. So all of them are very important, especially glycine cleavage system, most frequently asked, and the clinical correlations to Hartmann's disease, blue diaper syndrome. If you have any queries, you can send them before we end the class. Okay, thanks for watching. See you in the next class.